Hello, everyone, and welcome to today's broadcast, Destabilization and Degradation of MYC, More Ways Than One, presented by Dr. Jason Melo, Post-Doctoral Fellow at Princess Margaret Cancer Research at the University of Health Network. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that Ask a Question box located on the far left of your screen. Type the questions into the drop-down box that appear on that screen, and our speaker will respond to your questions via email. If you have any trouble seeing or hearing this presentation, just click on the Ask a Question box and let us know you're having a problem. This presentation is educational and thus offers educational credits. Please follow the Continuing Education Credits tab located on the top right corner of your presentation window and follow the process for obtaining your credits. Please join me now in welcoming our presenter, Dr. Jason Melo. I will now turn the presentation over to him. Welcome, sir.
Thank you, Susie, and thank you for everyone at LabRoots and the organizers of the Cancer Research and Oncology event for providing me with this presentation opportunity. As alluded to in the title, I'll be discussing some of our work within the Penn lab, studying the regulation of the MYC protein and its therapeutic implications within cancer. Before I get started, I just wanted to kind of highlight what I want everyone to get out of this presentation today. First off, I would like the attendees to get an idea about gain an appreciation for the critical role MYC plays within tumorigenesis. Also to develop an understanding of how the amount of MYC protein within a cell helps dictate the fate of the cell, and also understand how little we truly know about how the stability of the MYC protein is regulated and why this has hindered our ability to develop a therapeutic strategy against MYC. So to start off, what is MYC? The MYC protein, which is also known as c-MYC, is a member of the MYC family of transcription factors. The other family members are N-MYC and L-MYC. All MYC family members act as transcription factors that bind to DNA at conserved regions termed E-boxes. These E-boxes tend to be within promoter regions of DNA, promoter regions and enhancer regions of DNA. However, non-canonical MYC binding sites in the genome have also been identified. N-MYC, L-MYC, and C-MYC share much sequence homology, but they differ in their expression profiles. N-MYC and L-MYC are primarily expressed during development and were identified in neuroblastoma and lung cancer, respectively. C-MYC, however, is expressed throughout development and in adult tissues, and its increased expression is associated with many cancers. So what role does MYC play within normal cells, and why is it so important? Many of the genes regulated for MYC are important for cell growth, metabolism, proliferation, all of which are important for a growing cell. Unsurprisingly, the amount of MYC in the cell is correlated with the ability to divide. As the amount of MYC increases, it reaches a critical threshold where the cell is able to enter a proliferative state. At the proliferative state, the cell is able to divide. However, there's also a second threshold, which is an apoptotic threshold. If the amount of MYC in the cell reaches this point, instead of this is potentially oncogenic. And instead of normally dividing, the cell will end up going through apoptosis and dying. However, during tumorigenesis and cancer development, secondary mutations within apoptotic machinery, such as DNA damage response, that prevent the cell from undergoing apoptosis are gained, and this allows the cells to maintain MYC at oncogenic levels. This allows the cell to divide and proliferate uncontrollably. But in normal cells, the expression of MYC is tightly controlled. And so what we want to know is, how do cancer cells increase the amount of MYC levels to allow them to proliferate uncontrollably? We know that MYC is a key driver of tumorigenesis. Many cells acquire amplifications of the MYC gene. Roughly 20% of all cancers have amplifications or increased numbers of copies of the MYC gene beyond the typical 2 associated with a normal cell. However, despite its importance within tumorigenesis, MYC is surprisingly undermutated. There's only one known mutation within MYC that acts as a driver for cancer. However, while 20–30% of cancers have genetic lesions within the MYC oncogene, 50 to 70% of cancers have dysregulation of the MYC protein and increased expression of the MYC protein, underlying alternative mechanisms to increase MYC to allow a cell to divide uncontrollably. So what other pathways are able to increase the amount of MYC within the cell? There are primarily three main ways. One is through mutations of other oncogenes such as PI3 kinase, Wnt, ERK, HER2 that can all lead to aberrant signaling, which increase the expression and regulation of the MYC protein. The aforementioned genetic lesions such as amplifications or translocations of the MYC gene to a different promoter will all increase expression of the MYC gene, while the one characterized mutation of T58, prevents the efficient degradation of the MYC protein. And finally, altered stability of the mRNA protein or post-translational modification of the MYC protein can all conspire to alter MYC levels. In many cases, multiple oncogenic events can converge to increase the amount of MYC in the cell, such as amplifications of the MYC genes, as well as mutations of upstream signaling processes such as PI3 kinase pathway mutations. The net effect is increased levels of MYC protein and increased activity as a transcription factor. Increased MYC activity as a transcription factor. So how does MYC function as a transcription factor? MYC binds to its binding partner MAX, forms a heterodimer, and then searches the DNA for MYC binding sites and E boxes. Once it finds these binding sites, it's able to recruit molecules to these regions of DNAs to alter the genomic landscape or increase expression. These can include histone acetyltransferases, such as GCN5, STAGA, TIP60, that acetylate histones, open up chromatin, and allow RNA polymerase II to come in and transcribe genes. Alternatively, MYC can recruit transcription factors and elongation factors such as PTFB to modify RNA pol directly and allow transcription to occur. Finally, MYC can recruit repressive elements such as G9A, GLP, or MIZ1 that methylate histones and contract DNA to prevent RNA pol 2 from accessing genes and allowing for active transcription. All these functions lead to a model of MYC where MYC is a general amplifier of gene transcription. MYC is able to open up and control the chromatin landscape and recruit RNA polymerase to allow more gene transcription to occur. Essentially, as the amount of MYC in the cell increases, more MYC is bound to chromatin and allowing more modifications and more transcription to occur. So how is this so important? Well, MYC binds to 10 to 15% of the genome. MYC is a very promiscuous transcription factor and is able to change a very large transcriptional program. This transcriptional program is involved in the growth, metabolism, proliferation, apoptosis, stemness, and migration of the cell. And all these are important factors for tumorigenesis. This also means that targeting MYC would be a great therapeutic strategy. If we can decrease the amount of MYC in the cell, decrease the MYC levels, we could potentially put the cell into a cell stasis state or where the cell would no longer grow. And this would potentially impact the 50 to 70% of patients that have increased expression of the MYC protein. The problem arises in trying to develop a drug against MYC. MYC is not an enzyme, and as a transcription factor, it's intrinsically disordered. Most small molecule inhibitors are designed against enzymes and structured regions of proteins, making MYC a poor target and when it's often referred to as undruggable. Alternatively, we can try and target the mechanisms which conspire to increase the amount of MYC in the cell. However, we know surprisingly little about how the MYC protein is regulated. My work focuses on trying to characterize how the stability of the MYC protein is regulated by post-translational modifications, also known as PTMs. The MYC protein has actually been shown to be heavily modified by post-translational modifications. To date, about 70 modifications have been identified through high throughput means such as mass spec or low throughput techniques such as mutational analysis. And here we can see a schematic as reported on the web tool phospho site. This is in line with the consensus that the MYC protein is tightly regulated within the cell. However, the only PTENs that have been characterized are threonine 58 and serine 62. As I mentioned, threonine 58's mutations were identified in Burkitt's lymphoma and represent the only known cancer-causing mutation in the MYC protein. The regulatory mechanism centered around T58 and S62 forms the only known characterized degradation pathway for MYC. In this pathway, MYC becomes phosphorylated sequentially as serine 62 and threonine 58 by mitogen-stimulated kinases such as ERK and GSK3 beta, respectively. This puts MYC into an active state, allowing transcription to occur. PIN1 and protein phosphatase 2A are able to then come in and work together to dephosphorylate serine 62, leaving MYC with a sole phosphorylation at threonine 58. This forms a recognition sequence for an E3 ligase, SDF-FBW7, which is able to interact with the threonine 58 phosphorylated MYC, ubiquitolated, leading to its degradation at the proteasome. While other E3 ligases have been reported for MYC, this is the only known regulatory pathway to date. So why is mutation of T58 tumorigenic? The deubiquitase USB28 works to counteract this ubiquitination and prevent MYC from being degraded. So why is the mutation of T58 so tumorigenic? The half-life of the MYC protein is about 30 minutes. That is, it takes 30 minutes for 50% of the MYC protein to be degraded under normal conditions. Mutation of T58 to alanine doubles the half-life to approximately 60 minutes. This corresponds to an increased amount of MYC in the cells and increased tumorigenicity. Essentially, you've doubled the amount of MYC within the cell. Putting it bluntly, more MYC equals more tumor growth. And we see that when we look at assays of tumorigenicity. Mutation of T58 increases the amount of, in this case, colonies we see on soft agar assays, which is a surrogate marker for tumor growth. However, as I mentioned, MYC has a half-life of 30 minutes, making it a very liable protein that gets degraded very quickly. The median half-life of all proteins within the cell is about 46 hours. If the GSK3 beta and FCF pathway was the only degradation pathway for MYC, we would expect a greater increase in the half-life of the MYC protein when we inhibit this pathway. So what else is degrading MYC when FBW7 isn't? This forms one of the main research themes for the Penn lab, trying to understand how the MYC protein is regulated in the cell, and conversely, how it is deregulated in cancer. My research project centers on this hypothesis, on the hypothesis that the MYC protein is heavily modified by post-translational modifications, as I mentioned, about over 70. Many of these PTENs may form part of another regulatory pathway for MYC that we can exploit to develop a therapeutic strategy to treat patients with increased expression of MYC in their cancers. To begin to address this, we first decided to focus on clusters of residues which have been shown to be modified by post-translational modifications. In the N-terminal transactivation domain of MYC, centered around MYC box 1 and MYC box 2, there are 6 lysine residues, 5 of which have been shown to be modified by post-translational modifications. We decided to mutate all of these to arginine and see what effect this would have on the MYC protein. In a soft agar assay, where the overexpression of MYC allows colonies to grow under anchorage-independent conditions, which is indicative of a cell growing in the tumor environment, we see that MYC is able to drive tumor growth or tumor cell growth in these TET21 neuroblastoma cells under these conditions. Mutating these 6 lysine residues to arginine dramatically decreases the amount and size of the colonies we see. Because these residues are spread over a wide region of MYC, we decided to separate these into 2 smaller clusters. The lysines around MYC-box1, which we term 2KRN, and lysine residues around MYC-box2, which we term 4KRN. These residues seem as a logical place to split this larger cluster. What we see is that the mutation of the 4 lysine residues around MYC-box2 has minimal effect on the ability of MYC to drive tumor cell growth, whereas mutating the 2 lysine residues in MYC-box1 phenocopies what we see with 6KRN and dramatically decreases the amount of tumor growth we see. These 2 lysine residues in MYC-box1, lysine 51 and 52, are side by side. Our initial hypothesis was that these 2 lysines would be redundant, that mutating either one would have no effect. However, this wasn't the case. When we mutate lysine 51, we see very minimal effect on the ability of MYC to drive tumorigenesis. However, when we mutate lysine 52, we see the dramatic decrease in tumor cell growth in these conditions, and which phenocopies what we saw with 6KRN and 2KRN. This implicated lysine 52 as a causative, as an important residue for MYC function, and the causative residue or mutation for that 6KRN phenotype that we were seeing. We decided to take this into a xenograft assay. In this assay, we transduced Rat1A cells with MYC or our mutants of interest. When these cells are grown subcutaneously to non-skin mice, the cells that overexpress MYC are able to develop very large tumors very quickly, in contrast to the cells which do not carry any MYC, or the empty vector cells. Deletion of MYC-box2, which is a known loss of function mutation for MYC, is unable to drive this tumor growth phenotype that we see with MYC. When we look at our 2KRN, K51, and K52 mutants of interest, we see that 2KRN and K52 form very small tumors, whereas K51 grows a large tumor, very much like MYC, wild-type MYC. This, again, adds further evidence to implicate that K52 is the causative mutation leading to this loss of function phenotype, and it might be important for MYC function. These mutations are mutating lysines arginines. While this is a relatively conserved change, this may have a structural change under the MYC protein. Again, the MYC protein is intrinsically disordered and has very little secondary structure. However, we wanted to make sure that what we were seeing was due to a functional change and not a structural change. Our collaborators in Sweden, Dr. Maria Sunohargen and Vivian Morand, used circular dichromatism spectroscopy to assess the secondary structure of the MYC protein. This technique uses circular polarized light to assess the absorption of the protein over a range of wavelengths, allowing us to assess changes in secondary structure, specifically beta sheets and alpha helices. What we observed was that there was no difference in secondary structure after mutating lysine 51, 52, or both of them together, indicating that the changes we were seeing was due to a functional consequence of the mutation and not a structural consequence. So why is lysine 52 so important? It resides in a highly conserved region of MYC termed MYC-box1. And lysine 52 and 52 and the surrounding residues are 100% conserved across all MYC orthologs, including N-MYC, L-MYC, and MYC orthologs for species as divergent as chimps, rabbits, cats, fish, and frogs, indicating that there's an evolutionary reason for keeping these lysine residues around. Moreover, when we look at phosphocyte plus, we can see that lysine 52 has been shown to be modified by some PTMs. Specifically, these PTMs appear, lysine 52 seems to be modified by ubiquitination and sumylation. However, the consequence of these modifications is still really under investigation and is very unclear at this point. When we were doing our experiments, we noticed that the expression of 2KRN, K51, and K52, specifically 2KRN and K52, was much lower than our wild-type MYC or the MYC mutants. We didn't know if this was due to an expression issue or this was due to a functional consequence on the protein. When we looked at the mRNA expression, we saw no change in the mRNA expression of these proteins. What typically happens during a protein life cycle is that it expresses mRNA, it gets converted into a protein at the ribosome, and eventually it gets polyubiquitinated by an E3 ligase, leading to its degradation. And lysine residues are known to be modified by ubiquitin, leading to a potential degradation phenotype. So we wanted to see if this was due to proteasomal degradation. We treated the cells with MG132, an inhibitor of the proteasome. We're able to prevent proteasomal degradation and look at accumulation of the MYC protein. If the decreased stability of lysine 52 was due to increased proteasomal degradation, this should rescue the protein levels. And this is indeed what we see. Treating with MG132 rescues the stability phenotype, or the protein levels of the 2K-RN and K52 within the cell, indicating that lysine 52, or mutating this to an arginine, increases the amount of proteasomal degradation we observe. To look at this in another way, we want to see the stability of the MYC protein after mutation of lysine 52. When we treat the cells with cyclohexamide, an inhibitor of protein translation by targeting the ribosome, we prevent the conversion of mRNA into protein. We are then able to monitor how long it takes for the MYC protein, the remaining MYC protein within the cell, to be degraded at the proteasome. What we see is that after treating the cells with cyclohexamide for 0, 15, 30, 45, 60, or 90 minutes, we can see that the half-life of the MYC protein is about 30 minutes, meaning that it takes about 30 minutes for 50% of the MYC protein to be degraded. When we look at the 2K-RN mutant, we see that the half-life drops to about 21 minutes. Mutating K51R alone has minimal change on the half-life of the MYC protein, has about 27-minute half-life, whereas mutating lysine 52 to arginine again replicates what we see with 2K-RN and has a half-life of about 21 minutes. This indicates, and it adds further evidence to the idea that mutating lysine 52 destabilizes the MYC protein by causing increased proteasomal degradation. As you mentioned, the most well-characterized degradation pathway for MYC is through SCF-FBXW7. Lysine 52 is very near threonine 58 and S62 within MYC box 1. There was a potential that by mutating this lysine residue, we were having an effect on the canonical degradation pathway for MYC. So we used two independent approaches to assess this. We first looked at phosphorylation and saw no change in phosphorylation. Then we used CRISPR-Cas9 genome editing to knock out SCF-FBW7. In our wild-type cells, as we saw with the previous slide, the half-life of MYC is about 30 minutes, and the K52R half-life is about 15 minutes, or about, 18 minutes. When we knock out FBXW7 using CRISPR-Cas9, we increase the half-life of MYC to approximately 63 minutes. And the K52R increases to about 34 minutes respectively. But tellingly, we still see a dramatic difference in the amount and the stability of lysine 52 mutant. If this was due to FBXW7, we would expect the half-life of K52R to be rescued and to be around 60 minutes. Seeing that we still see a differential in these cells, it would indicate that the degradation pathway is independent of FBW7. It still exists even when we knock out FBW7. As an independent approach, we took advantage of the netylation inhibitor MLN4924. Cullen ring ligases such as SCF-FBXW7 rely on netylation of the Cullen backbone in order to remain active and able to polyubiquinate and degrade its substrates. When we treat with MLN4924, this inhibits activation of NET8 and prevents netylation of the Cullen backbone. So we use this to as an independent way to assess if FBXW7 was important for degradation of the lysine 52 mutant. When we treated cells with MLN4924, we see that the netylated Cullen 1 disappears after treatment. We also see increases in the other known target of SCF-FBW7, cyclin E. When we look specifically at what's happening with MYC, we see that after treatment with MYC, or with the MLN 4924, we see an increase in the amount of MYC protein, roughly a doubling. When we look at the T58A mutant, we don't see any change or any noticeable change in the amount of protein in the cell, as this is already protected in this pathway by mutating this key threonine residue. When we look at 2KRN, we see an increase, but tellingly it doesn't reach the same levels as MYC or T58A. It's not rescued. K51R reaches those levels we see with MYC and T58A, and K52R does not. This is in contrast to what we saw with MG132. If we inhibit all proteasomal degradation, we're able to completely rescue the levels of MYC in these cells. Whereas if we treat with MLN 4924, which only inhibits colon ring ligases such as SCF, FBXW7, we don't see that effect. We don't see that complete rescue. This leads to us putting together a working model. In this model, we suspect that MYC is unstable when it has no post-translational modifications. In our model, a lysine modifying enzyme will come in and add a post-translational modification, potentially ubicurylation, sumylation, acetylation to lysine 52, leading to the recruitment, allowing stabilization and maintaining MYC in an active state, allowing it to interact with other protein-protein interactors. However, if no PTEN is added to lysine 52, MYC is unstable, leading to its polyubicurylation and degradation by an E3 ligase. In this model, the lysine modifying enzyme, which adds the post-translational modification onto lysine 52, would be a very promising therapeutic target to develop a strategy to target MYC in cancer. Our goal is now to identify the E3 ligase, the lysine modifying enzyme, and the interacting partners that keep MYC in an active state to identify therapeutic strategies and put together this entire degradation and regulatory mechanism. To do this, we're taking advantage of BioID. BioID is a technique where a board of biotin ligase is fused to your protein of interest, in our case MYC. This biotin ligase, when it's able to activate biotin but releases it in proximity of your protein interest and actually tag any proximal proteins or protein-protein interactors. So when we express these in cells, the MYC protein will interact with its interacting partners. When exogenous biotin is added, this will tag the protein-protein interacting partners, which we can then harvest and purify using streptavidin beads and send for mass spec analysis to identifying those protein interactors. Our lab has previously used this to identify the MYC interactome and fully characterize new interacting partners for MYC. My approach has been to now take this a step further, and in addition to doing the wild-type MYC interactome, look at the MYC interactome for the 2KRN, K51R, and K52R mutants, and identify their individual interactomes and compare them to one another to see what changes occur in order to identify any potential therapeutic members of this degradation pathway and therapeutic targets. This strategy is based on some of the information and the data we've developed. We know that 2KRN and K52R both have a shorter half-life and reduced ability to transform cells, likely meaning that they are acting in a very similar way. Mutation of lysine 51 has a normal half-life and transformation potential seems to be unaffected. Therefore, if we look at the individual interactomes and we focus on those changes in the interactome that are unique to 2KRN and K52R, but are independent of changes in the K51R interactome, we may be able to identify these different members of this degradation pathway. We've recently done this work. We've collected the interactomes of each of these proteins and done this analysis. We have actually identified an E3 ligase that we suspect is increased in our lysine 52R mutated samples that we're trying to assess more fully. We found the lysine modifying enzyme, which decreased in our mutant, which we suspect is because it's no longer able to interact with this and modify the MYC protein, that it might be important for this pathway. We've also identified changes in protein complexes that interact with MYC and are important for MYC function. Right now, our work is characterizing these different approaches to help identify this regulatory pathway and hopefully develop a strong therapeutic strategy we can potentially move into the clinic. So in summary, our learning objectives, my learning objectives for this course or for this session was to learn about the critical role MYC plays in tumorigenesis. We know that MYC binds at 10 to 15% of the genome as an important transcriptional program that helps mediate tumorigenesis, primarily by pushing the cell into a growth and proliferative phase. I wanted to get across the understanding that the relationship between the amount of MYC and the protein in the cell has a direct impact on its tumorigenicity. As the MYC reaches a critical point, the MYC levels within the cell can enter a proliferative stage. However, too much MYC results in an oncogenic state, which under normal conditions would trigger the cell to undergo apoptosis and die. However, cancer cells, which acquire mutations in these apoptotic pathways, are able to tolerate high levels of MYC and able to proliferate uncontrollably. Finally, I want to understand how little we actually know about how the MYC protein is regulated, how its stability is regulated in the cell, and how this has hindered our ability to develop strong therapeutic strategies against MYC. We know about the GSK FBW7 degradation pathway, but now we have a potential new pathway which we can develop new therapeutics against it. So I'd like to thank everyone, all members of the Penn Lab, my supervisor, Linda Penn, and our collaborators, Dr. Brian Rout and Dr. Maria Sunohagen. And what questions do you have?
Thank you, Dr. Damu, for that informative presentation. And we will now start that Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window. Type the question into the box that appear on the screen and click that Send button. Our speaker will follow up with you via email. So here are some questions from our viewers right now, Dr. Damu. Mutation of lysine residues is typically associated with the stabilization of a protein by preventing polyubiquitin, I always have a hard time with that word, polyubiquitination and degradation. Why does mutation of K52 destabilize MYC?
That's a great question. So it's true that typically when you mutate lysine residues, you see stabilization by preventing polyubiquitination. However, what we see with K52 is the opposite. We see a destabilization of MYC. With polyubiquitination, the degradation mechanism is typically associated with lysine 48 and now lysine 11 linked ubiquitin chains. This is recognized by the proteasome and triggers degradation. However, the other lysine modifications that can occur, such as lysine 63 ubiquitin chains, sumylation, acetylation, are not necessarily associated with degradation. This is why we suspect that the modification that's occurring on that lysine 52 is actually not a lysine 48 ubiquitin chain or lysine 11 ubiquitin chain, but one of these other less known or less characterized lysine modifications.
Thank you for that answer. And are there any other E3 ligases reported for the MYC protein?
Yeah, there have been a number of studies reporting ligases that are able to degrade MYC E3 ligases. These include beta TCRP, SCIP2, QE1, ELL, to name a few. These have been mostly one-off studies that have identified an E3 ligase as important for degrading MYC, but not necessarily one that, or a degradation pathway that we can exploit. And also it's important to note that targeting an E3 ligase would not be a viable therapeutic approach. We want MYC to be degraded, but we actually want that E3 ligase to be active. So what we need to find is the other components of that degradation pathway that are important for its degradation. And what's interesting to note is that in our data where I showed treatment with the MLN4924, which is that inhibitor of all colon ring ligases, beta TCRP, SCIP2, these are other colon ring ligases that are also inhibited in those settings. So So that data also implicates a non-culinating ligase in this degradation pathway. So it also gives us some further evidence to move on.
Thank you. And a final question for you, Dr. Melo. Why haven't any therapeutics against MYC been developed?
We've known about MYC for about 30 years. It's been a very challenging protein to work with historically. It's buried within the chromatin. It's liable. It degrades very quickly. Has no very good structure, and our best therapeutics in small molecules to date have been all designed against enzymes that have very defined enzymatic pockets that are easy to target. So MYC has been one of those proteins that has been considered undruggable to date. There has been a lot more recent success using inhibitors such as JQ1 and the BRD4 inhibitors that target BRD4 but don't directly target MYC, but have a dramatic impact on the expression of MYC. So again, by decreasing the amount of MYC in the cell, it seems to be a therapeutic vulnerability we can take advantage of cancer. So as we're starting to learn more about how MYC is regulated and how MYC is expressed in the cells and how different ways that we can decrease the amount of MYC in the cells, we have a much better chance now of developing these therapies.
Dr. DeMello, thank you for your presentation today, and thank you for your important research. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 2019. You'll receive an email from LabRoots letting you know that this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now, and thank you for joining us. We hope to see you again soon. Bye-bye.